Precision Cancer Care. I'm Dr. Nathan Goodyear, Medical Director here at Brio-Medical in the beautiful Scottsdale, Arizona. When you look at the treatment of cancer over the last roughly century, 100 years, it's been a strategy to treat a one-size-fits-all kind of approach. And that is an approach, that is a process that has failed. Now, how can I say such a thing? Because when you look at this process of cancer, the medical industry, the world as a whole, has been going to war on cancer for well over 50 years. President Nixon declared war on cancer in his presidential announcement State of the Union in 1971. But it was ongoing even before that. When you look at one of the standards, one of the pillars of conventional cancer treatment, it's chemotherapy. That was born out of World War I into World War II. So this concept of going to war on cancer has been going on a long time. But it's been structured as a one-size-fits-all approach. And it's structured out of the kind of magic bullet theory. This was a theory that was first described via Paul Ehrlich in the late 19th century, kind of looking at the process of how disease developed and in the treatment of syphilis. Now, that process, that concept, that paradigm of a one-size-fits-all approach, magic bullet, it's dead. It does not apply anymore. Our understanding, because of science, really helps us to see that this is individualized. To be successful in the treatment of cancer, we need precision. Now, intuitively, as humans, we recognize this because we can have 5 patients sitting in our lobby. One will be 6'5", 350 pounds. Another one may be 4'8", 118 pounds. Intuitively, one has prostate cancer, one has pancreatic cancer. Intuitively, we recognize the difference. But yet, conventional medicine in the paradigm today, and for the last 50 to 100 years, has said, no, let's one-size-fits-all approach. Now, I said that the data is telling us this hasn't worked. And I've referenced this study several times. It's called the PURE study. That's the acronym for it, the Prospective Urban and Rural Epidemiology Study, where they looked at countries based on income. And they found that higher income countries were seeing a massive spike in cancer. As a mortality perspective, they found that in those high income countries and even the higher middle income countries, cancer was now the number one, mind you, cause of mortality in adults over cardiovascular disease. And in fact, when you look in the United States, 13 states, as of current data, the cancer is the number one cause of mortality over cardiovascular disease in adults. And it was first described in Alaska many years ago. So this is not a new phenomenon, but it's been one that just doesn't seem to get into the media, because cardiovascular disease as the number one cause of mortality in adults has just kind of seemed to be the presumptive leader, but in actuality, cancer is the leader. And when you look at the spike proteins, whether it's via injection or infection, that only seems to be aggravating that issue, and we'll see that moving forward. Literature describes that. So we're not winning this war on cancer, so there needs to be a paradigm change away from the one-size-fits-all approach, the magic bullet. We need to move to precision cancer care. So how do we do that? Specifically, how do we do that here at Brio-Medical? First, well, it's 5 ways. First is genomics. Genomics is just the collection of the genetics of the organism. Now, this could be the collection of the genetics of the organism, that being a human, but also it could be the genomics of the cancer. And understand, within the context of a person with cancer, we have the genomics of the individual, but we also have the genomics of the cancer. And those can be very different. And so when you look at the genomics, this is the DNA. And when you look at cancer, there is genetic mutations that often are associated with cancer. For example, P53. This is a tumor suppressor gene that is turned off in well over 60% of all cancer types and varieties. There was a recent study that came out that showed that a Western diet high in animal protein, resulted in a shift in the gut microbiome and specific bacteria, particularly one called a PK3 plus E. coli bacteria. It produced a metabolite that actually caused direct DNA damage, a genomic alteration. So that study here just published this month showing how nutrition can impact the genome that could be a part of cancer. In this case, it was in colorectal cancer. Likewise, you can eat fermented foods and increase Lactobacillus reuteri, another type of bacteria in the gut, and increase another metabolite, reuteri, and this can actually inhibit proliferation in vivo and in vitro studies. So again, really seeing that diet through the gut bacteria can actually be a means to start the treatment of cancer. That's genomics. Powerful therapy, very precise. And in the future, what I think we'll actually see is actually customized probiotics, customized therapy to target the gut microbiome, the gut flora, the gut bacteria, to really not just help other therapies work better, but to begin the initiation of healing in the gut, but that permeates throughout the rest of the body as a therapy in and of itself. And you've actually seen this with what's called fecal transplants for things like ulcerative colitis. So I think this is the future, this is the targeted, and this is how we can actually affect genomics in cancer, or for just the regular genomics within the body that's not related to cancer, all through diet. That's just one example. Number two is epigenomics. So there's genomics, that's the foundation, that's the DNA, the collection of all the DNA in the organism, and as it relates to cancer, it can have a separate epigenome. Epigenetics is the expression above. So DNA gets transcribed into RNA, and that then gets translated into protein. And epigenomics is everything above the DNA. So in essence, it's the DNA being transmitted into action. And that's really what epigenetics is. So it means literally above genetics. And so when we look at epigenetics, this is a lot of what's happening in cancer. Just as you have genomics, you also have epigenomics. You can have it of the individual, and then there'll be separate epigenomics as it relates to the cancer. So here we see massive epigenetic shifting, changes in expression as it relates to cancer. Now, normally changing the genome can take thousands of years, generations, but epigenomics, it can change like that. And then it can be inherited down to offspring. So ways to attack epigenetics are also another form of precision cancer care. So how would we do that? Well, studies have also shown nutrition can be used to alter estrogen metabolism through epigenomics. For example, broccoli. Broccoli and what are called microRNA coming from broccoli, which are small pieces of RNA which come from DNA through transcription. Small pieces are called micro. These can actually alter estrogen metabolism in a way that reduces breast cancer risk through poor estrogen metabolism via what's called 4-hydroxyestradiol and 4-hydroxyestrone, which are very, very carcinogenic species because of how they bind to the DNA irreversibly in many ways and cause DNA damage. Vitamin C can also impact epigenomics in the cancer to counter the epigenetic changes that promote cancer growth, survival, and spread. Those are just a few examples of how we can, through genomics and epigenomics 1 and 2, target and provide precise cancer care here at Brio-Medical. Number 3 is transcriptomics. I talked about how DNA gets encoded into messenger RNA through transcription. Transcriptomics are just the process of looking through all the different RNA that can be a byproduct of that process of transcription, which is an intermediary step to proteins from translation. Now, this includes messenger RNA. Of course, the last couple of years, I think everybody's an expert on messenger RNA. There's, fragmented RNAs, microRNAs, ribosomal RNAs. There's many different types of transcriptomics. And this is an intermediary signal step that we can test for. We can provide precision treatment to affect a change. Both genomics, epigenomics, and transcriptomics, all of these we can evaluate, and we can, in essence, provide precise assessment and thus precise treatment based on these testing. This can be testing of not just the tumor via histology and pathology, but also via blood. And through this, we can then provide precise assessment and precise treatment plans. So as it relates to transcriptomics, our treatments are going to dive deep into this process of affecting all these transcription products. And I already mentioned, for example, the one which is broccoli. Mom told you eat your broccoli. Mom knows best, right? But little did she know that this was related to actually transcription and transcriptomics, which is a relatively recent scientific genre, of the classification broadly of omics. And this is the future that's present now, the omics level of cancer research and treatment. But broccoli, again, microRNA is going to affect transcriptomics. High-dose IV vitamin C, curcumin, quercetin, melatonin, all of these are examples of how we can specifically affect transcriptomics, transcription process, to change how DNA, whether it's in a normal healthy cell or whether it's in a cancer cell, we can change the expression of that through altering transcriptomics. So not just testing and evaluating, we can test and go, wow, we can affect a change. Another really exciting example that we're involved with research here at Brio-Medical is peptides. Peptides are short proteins, typically less than 30 or 50 amino acids. Beyond 50 amino acids, these amino acid chains are called proteins, but less than that, they're classically called peptides. A relatively new arena as it relates to precision cancer care, but there are targeted peptides and then there are what we call custom peptides. And here specifically, I'm talking about developing customized precision peptides based on tumor assessment of proteomics, based on blood assessment of transcriptomics, allowing us to develop precise peptides to actually target the tumor, but also stimulate the immune system to do its job. Novel concept, precision evaluation, precision treatment. The fourth is what's called, first is genomics, second is epigenomics, third is transcriptomics, the fourth is proteomics. Proteomics is proteins. So proteins come from initially the DNA, then through transcription, then to translation. So proteomics is just the study of the entirety of the protein in an organism in healthy or non-healthy, here, cancer. So collectively looking at all the proteins that are being expressed is what proteomics is. And what we want to do is not just look at them through testing, as I mentioned, in the tumor or in the blood, but it's also to assess for the effect of treatment. And here, same way, through these precision customized peptides, we can see what the tumor is expressing, where the body's expressing, particularly as it relates to the immune system, and developing a strategy that alters the proteins in the proteomics in the cancer in a way that inhibits cancer growth or counters it, but also stimulates the immune system, natural killer cells, cytotoxic T lymphocytes, and in the tumor microenvironment, so that we can shift the balance in the tumor microenvironment throughout the body so that there's more healing, there's more anti-cancer than there is promotion of cancer. So that's step number 4 of precision cancer care here at Brio-Medical. Step number 5, and the last, I think is the most exciting and one of the more newer areas. And it's really I think the area where we can have the biggest impact. It's called metabolomics. Metabolomics is just simply assessment of the entirety of metabolites. Metabolites are a series of step down, basically detoxification products or altered products that the body will take, hormones, neuropeptides, anything in the body. It's how the body will change and alter these proteins in the body. So, for example, when you look at hormones, there's estradiol, estrone, and you have metabolites, 2-hydroxyestradiol, 2-hydroxyestrone, 4-hydroxyestrone, 4-hydroxyestradiol. Testosterone, you have metabolites of 5-alpha dehydrotestosterone and 3-alpha androstenediol. Cortisol, you have tetrahydrocortisol. The point here is you have the parent compound, and then you have metabolites of them as enzymes add to or take away. Now, it used to be that we looked at metabolites as kind of being inert, metabolically inactive, but we now know that many of these metabolites are not only metabolically active, they could be metabolically more active than the parent compound themselves. And metabolomics is just the evaluation of the entirety of the metabolites in the body. Not just for the purpose of evaluation, but for the purpose of generating a precise cancer care treatment program to affect metabolomics against cancer. And I've already mentioned many ways already that we can affect metabolomics. Talked about broccoli. You know, many people are advocating taking, supplements, particularly broccoli seed extract, to affect estrogen metabolites. That is a metabolomics perspective. And when you look at the things and the treatments that we can offer here at Brio-Medical, all of these are working in the metabolomics perspective. In fact, I was sitting at one of my favorite coffee shops doing some writing, which I do on a regular basis, and there was a gentleman sitting next to me, and he happened to overlook me, my shoulder at my laptop when I was working on a presentation I gave at Denver here recently. And I was in this portion of my lecture creating slides dealing with vitamin C and metabolomics. And there's actually several different studies and review articles written on just that topic of how high-dose IV vitamin C is affecting metabolomics in the cancer cells, very different than healthy cells. And he was a PhD recent graduate in the field of metabolomics. And so we got into this very interesting discussion about his area of research and vitamin C and how this exciting kind of marriage is coming together in this arena. So when you look at high-dose vitamin C, again, this is different than the oral antioxidative effects of vitamin C. High-dose IV vitamin C is going to be pro-oxidative. And what metabolomics research has shown is that with high-dose vitamin C, what it's going to do is going to create an energy crisis in the cancer cell, and it's going to create a detoxification crisis in the cancer cell. And it does this via two ways. Number one, it's going to deplete active glutathione, reduce glutathione in the cancer cell. And this creates the detoxification crisis, allows overwhelming oxidative stress within the cancer cell, which it can't handle, depletes catalase, which is a detoxification enzyme. This triggers what's called apoptosis, and the cancer cell dies. But the energy crisis, I think, is more important here, because what happens with high-dose vitamin C, it actually triggers a decoupling of what's called the pentose phosphate pathway from glycolysis, and this in turn leads to a depletion of NAD in part, and that causes the energy crisis, which changes the cell membrane polarization and triggers its cancer cell death. So when we look at the future of cancer care, precision cancer care, it's a change in paradigm in how we're going to approach not just the target of tumors, but the healing of the body. And I've mentioned several different ways, but there are 5 primary methods and treatments that permeate through all of them: genomics, epigenomics, transcriptomics, proteomics, and metabolomics. Here at Brio-Medical, we're not just going to evaluate those, in the tumor, in the blood, in your immune system, but we're going to create targeted, precise treatment strategies based on those assessments to not only target the tumor, but heal the body, particularly the immune system. That's the future of precision cancer care that's present now at Brio-Medical. To learn more about precision cancer care here at Brio-Medical, I encourage you to go to our website at brio-medical.com and go through our website and find the blog post, all the information there that talks about precision cancer care. Look forward to seeing you and developing and working with you in your precision cancer care treatment program in the future. I'm Dr. Nathan Goodyear, Medical Director here at Brio-Medical in Scottsdale, Arizona.